the people on the computer for consent. Okay, so the membrane and membrane transport. The extracellular matrix is the complex array of macromolecules attached to the cell membrane. It includes glycoproteins, which are carbs and proteins. An example is collagen. Proteoglycans are a type of glycoprotein that's mostly carb and just a small bit of protein. These are very fibrous. The fibronectin connects the extracellular matrix to the cell membrane, and then integrins are the large anchor proteins that connect to fibronectin. And they span, or they go all the way through the cell membrane. So here's if this is your cell membrane, there's the integrin, the fibronectin is attached to the integrin, and then the glycoproteins are attached to that fibronectin. The functions of the ECM are to regulate cell function, cell signaling, and to create cell junctions. The first type of cell junction is a plasmodesmata. It's only found in plant cells. This is a membrane-lined channel filled with cytosol. It allows water and small molecules to pass between plant cells. So here is the plant cell wall of one cell and the plant cell wall of the second cell. There's the plasmodesmata in between them. A tight junction is the plasma membranes of neighboring cells are packed tightly together, and it prevents linkage of fluid. An example of this is skin cells. As you can see here, they're really tightly packed together. That's so that water from the outside and water from the inside can't cross that really thick layer of cells. Okay, we'll give them a second to get back from the bathroom. I can crop this out. Okay.
That's what was in the runoff from those bashful.
Did you do that? Oh, I don't think I did because I didn't figure it out.
Yeah, I didn't vote for me.
Next.
Why am I most dependent?
I don't know.
Exactly. Most bashful Kessler Eagle.
Okay.
I hope he does. Because Kessler not one.
Kessler didn't get my vote because he sat here and said, I guess I'm doing the call. So, thank you.
Okay. We are continuing, so y'all listen up, please. Okay, the third type of cell junction is a desmosome. Desmosome. So this is an anchoring type of junction, meaning that it's basically got points where it's anchored from one cell wall or from one cell membrane to another, and at that junction it works like a hinge. So it functions like rivets, fastening cells together into strong sheets. So it basically allows for movements at the point of those rivets, or it doesn't allow for movement at the point of those rivets, but outside of those rivets it can move. So like I said, think of it like a hinge, or if you don't know what a rivet is, it's just a point that something can spin and twist and move on, but it's still connected at that point. Can anybody think of a type of cell in our body that would need this type of junction?
I'm guessing there's an example in the book.
There's an example in the book. But I just wanted to see if you can think of it. So what type of cells do you think would need to be connected at a point but still need to move and adjust? No.
You're wrong.
Close to bone. Do it. So not so much the actual joint itself, but muscle. Exactly. So like your muscle is attached at a point, but then other than that point, that muscle is able to move and twist and do what it needs to do. So an example of this is muscle cells. So again, these muscle cells are attached at this rivet, but then they're able to move in all sorts of different directions outside of those rivets. So like a muscle cell, this muscle cell isn't attached to the next muscle cell, only at this rivet is where it's attached. Other than that, it can move alongside it. If our muscles weren't like this, then you wouldn't be able to like stretch your arm out because those muscles would be attached at every point along that cell membrane. And so they wouldn't be able to stretch and move and do what they need to do.
So thought out.
Yeah, come here.
Okay, and there's one last type of cell junction, and it is a gap junction. So this is known as a communication junction. It provides cytoplasmic channels. So this is very similar to a plasmodesmata, just minus the cell membrane or the cell wall. So it basically connects two cells just like a plasmodesmata, just without the cell wall. So this is cell 1, this is cell 2, this is your gap junction. So it's called a gap junction because it's quite literally a gap in the cell wall. It allows for easier movement. We'll look next into like membrane permeability and like what's allowed to be let in and out of the membrane and why. And so this takes that out of account. So it's able to let larger molecules through that might not be able to move through the membrane regularly. Examples of this, can anyone just think of where that would be useful for one cell to need to basically dump nutrients into another? So an example is embryonic cells. So like in umbilical cords.
Oh, we don't need those anyway.
You need them while you're being formed. Okay, so basically whenever there is a fetus, that's such a weird word, whenever there's a baby inside of a stomach and it's developing, there's need for a lot of nutrients in high volumes being transferred from mother to baby. And so those cells are quite literally connected to one another via these large gaps, so it can dump those nutrients where it needs to go. There's not as much regulation between cell to cell like there is in your normal cells. So anyways. Okay. Does anyone have any questions over the 4 different types of cell junctions? No?
I feel like it's about to be a lot more complicated.
No. It's really not. Membrane transport's actually really easy to think about. Okay. So the membrane itself, what do you think it means whenever it refers to the cell membrane as a fluid mosaic model? What do you think that means? So what does fluid mean?
It's liquid.
Liquidy. So that's mainly referring to the way that it moves. So it's fluid in its movement. What does mosaic mean? No, that's Mozart. Okay. So does anyone know what like a mosaic painting or a mosaic thing is?
Like that weird tile thing?
So yeah, so it's like whenever people like can break up tile and then combine it to make it an image. That's a mosaic. So it's basically bits and pieces that are put together to make one big unit. So that's what the membrane is. It's basically a bunch of little bitty bits and pieces that are put together to make this one big functional unit. And that unit is very fluid in its movement. And so that's why it's called a fluid mosaic model. So we talked about a little bit about the ECM and how those integrins are pieces of proteins like within this membrane. Well, that is not the only thing in a membrane. So I'm basically just going to draw and label this membrane, and then we'll talk about these parts. So it'll be just a second here. And I'm trying to draw this as like big and obvious as possible.
Why does it look like it has arms and legs?
Because it's not done yet.
Irritating.
It looks like a what?
Ever seen Toy Story?
Yes. Who hasn't seen Toy Story?
Uncultured people.
I agree.
That it was tater time.
Our nose are lipid.
These are lipids. Look at you, remembering what a lipid is.
I didn't actually have no idea.
Good. See, y'all remember stuff. If all you remember from 10th grade is that this is a lipid bilayer, then I am happy with that. Okay. That is a very ugly lipid bilayer, I feel like, but okay. So if this is your cell membrane, this is a phospholipid. So one single unit of this is a phospholipid. Of each of those phospholipids, there is a hydrophilic and a hydrophobic portion. So which portion is hydrophilic? The one that likes water. The outside part. So the circles, good. So this and this are hydrophilic, meaning water loving. I'm trying to like keep it all on the paper, but also keep it zoomed in where you can see. And then the tail portion here is hydrophobic, meaning water hating. So that is quite literally like how a cell membrane is held together. That's it. It's just from its properties on whether it likes water or dislikes water. So the side that likes water associates itself with the aqueous environment inside and outside of the cell. And the inside hydrophobic tails situate themselves inside to create this barrier. That is the only thing holding them together. And again, these forces, these hydrophobic and hydrophilic forces are weak-ish forces. So they're kind of like hydrogen bonds. There's a lot of them, so it's a strong bond between water to water. Same thing. There's lots of lipids, so there's lots of reaction with the water around it. But the individual bonds themselves, or the individual reactions themselves, are pretty weak. And so that's what makes this fluid. So like our cells can situate themselves to the type of environment that they need to a certain extent. So like cells aren't always this rigid shape. They can change the shape in order to fulfill whatever need they have. And that shape is dependent on what is in the proteins. And so again, this is just an example of a protein. And because of this fluid mosaic model, so because of these weak reactions, this can move laterally to where it needs. So let's say there's a portion of the cell that is trying to get rid of, or not get rid of, but send a protein out to send to skin cells to add collagen to the skin cells to repair some damage. And that collagen is produced inside of the cell. So like that protein is spit out by your ribosomes. Well, that protein needs to get out of the cell. And so it might have to have a transport protein in the membrane to take it and send it where it needs to go. If that particular collagen fiber is on the other side of the cell, this can like move where it needs to go through the membrane. It can move laterally because of those hydrophobic and hydrophilic properties. Does that make sense? So it's kind of like, I know I keep saying things are conveyor belts, but it can kind of go back and forth like on a conveyor belt. And these proteins, they're very specific. We've already talked about like proteins being enzymes and how they're super duper specific on their functions. That's like all we've said so far. And that's kind of all you need to know at this point in time is that the function of that depends, or the structure of that depends on its function. So just generally what we've been talking about. So a membrane is also referred to as selectively permeable. Does anyone know or remember what that means? We talked about it in 10th grade, but it was a long time ago. It's something about water. So if something is selective, what is it? Like if you are a selective person, what does that mean that you are? Picky. So it basically you're choosy or you're picky on what you want or what you need or things like that. So the membrane is picky on what it lets in and out. So the permeable, that word permeable just simply means able to get through. And so the membrane is picky or selective on what it lets through. And I'll write that out in just a second. I just want to make sure everybody's gotten done with what they need. Okay, cool.
What they call permeable potato.
No. After I write this, I'll give you all a little break because I know y'all have been writing a lot. So it's selectively permeable, meaning it is selective on what it lets in and out of the cell. So it's very particular on things that it lets in and out. Water is one of those things that can pass through. What other types of molecules do you think need to pass in and out of the cell?
The ATP.
ATP, good. What else?
Protein.
Different proteins. Think smaller. What are you doing when you breathe in?
Oxygen.
Oxygen. So oxygen needs to pass through. What do you breathe out? CO2. So things like oxygen, CO2, water, different ions, all of these things need to pass in and out of a cell pretty regularly. So whenever you're breathing in, you're taking in oxygen, and then that oxygen has to go to every cell in your body in order to keep your organs alive. And so obviously oxygen has to be able to diffuse easily. I'm going to give you all like a couple-minute break, and then we'll talk about why that stuff can pass through and why stuff can't pass through. Yes. Okay, so before we start talking about what is able to go through a membrane, remember this statement right here. Like dissolves like. You're wrong.
Oh, no.
I'm just kidding. So, like, what that means is that if something is an ionic or it has a charge, it's going to react with something else that has a charge. Oh. No, sorry, that's a K.
What I
So,
said, I actually just wrote
like dissolves. That's funny. No, sorry, like dissolves like. So what that means is if you have a polar molecule, it's going to react with a polar molecule. Polar and nonpolar don't react with each other. That's why oil doesn't mix with water. That's why the hydrophilic heads are what's reacting with the water, and the hydrophobic tails of the phospholipid aren't reacting with it because it's polar going with polar. So remember that before we start talking about it. So basically, nonpolar molecules are able to pass through the membrane easily. So things like CO2, O2. These are really, really easy to pass through because they are not reacting with those hydrophobic portions of the tail. They're not being repelled by those. You might think, well, what about water? We'll talk about water in a second. So these nonpolar molecules, they don't need any sort of channel. They don't need any sort of protein. They don't need anything to move out and into the cell. Do not need proteins, channels, or pores to move in and out of the cell. They're able to diffuse in and out. Ow. And charged. So again, polar just means that it's got a partial positive and partial negative, whereas charged would be like the molecule itself has a negative charge or a positive charge. So what do you think about those molecules? So like water or sodium or chloride. What do you think about those? Oh, sorry. How do you think those get through? Good. So these get through with the help of, they're called transport proteins. So they take different forms. So they can be channel proteins, or they can be carrier proteins. So channel proteins work like a pore. Carrier proteins work like a door.
That rhyme. Was it meant to rhyme?
No, I just made that up. So channel proteins, like I said, it's like a pore. It's an actual opening via a protein in the membrane. So it's like this big protein here, just with like a channel in it. And it's got that cytoplasm inside of it. And so obviously if the ions are able to be in that cytoplasm, because the cytoplasm is polar, it's mainly water. So if they're able to react with that, then they can go through that pore because they're just using that channel. A carrier protein works like a door, so it's usually pretty specific to the type of molecule it lets in and out. Basically, think of a sodium molecule knocking on the sodium molecule door. That door opens, and then it shuts behind it. So it's just basically carrying that sodium molecule over where it goes. Excuse me. So basically, whether they're hydrophilic or hydrophobic, so nonpolar, again, hydrophobic, so they're able to pass through with the hydrophobic tails. Polar or charged molecules are hydrophilic, so they're going to stay where water stays, and they're going to get through the membrane how water gets through. So that's through channel proteins or carrier proteins. So that property, whether they are polar or nonpolar, determines what gets through. What do you think determines where it's going? So like in or out of the cell. We talked about this in great detail in 10th grade. This is when we did like the egg lab, and we talked about, what did we talk about with the egg lab? It's this fancy term that has to do with the movement of water across the membrane.
Oh, I remember.
Any takers?
Made it see-through.
You made it, yeah. So we dissolved the shell. Osmosis. Osmosis. Yay. Okay, this is the last thing we're talking about, and then we will have a practice problem. Not a AP College Board practice problem, but a different type of practice problem. Okay. This is still in membrane transport. This is just passive transport. So what do you think passive transport means? Do you remember? There's passive and active. If you're passive, do you think you're using energy or not using energy? Not using energy. Requires no energy to move molecules across a membrane. So one example of this is diffusion. So diffusion is basically exactly what you think about whenever you think of something spreading out in water. So if you drop food dye in water, it's all concentrated in that one drop, but then eventually after a couple seconds, the entire cup of water is going to be blue or green or whatever color that dye was. That's because those particles, the ones that are pigmented with color, are spreading out evenly within that water. And so that is an example of diffusion. It's just the movement of particles that spread out into available space. So if let's say there's a bunch of particles on this side, this membrane is permeable to those particles, then they will spread out. Here, I'm going to make it even to their available space. The whole goal is that these molecules just want an even amount of space so that they can sit there and exert their energy without bumping into any other molecules. It goes back to the whole property of that molecules want to be in their most random state. That space allows them to be in their most random state. So in the absence of any other forces, so if there's not anything creating that barrier, keeping that on this side, or if there's not any other molecules over on this side that it's trying to compete with, a molecule will go down its concentration gradient. So that means going from high concentration to low concentration. Molecules always want to be less concentrated because, again, that allows them to have their space to move around in. So what do you think if that barrier wasn't letting the particles through, but it let water through, where do you think the water would move to? So like ignoring this picture, if you have this picture and that membrane wouldn't allow those black particles to go through, what direction do you think the water would go? It would go here. to let those molecules spread out more. So that is what we talk about when we talk about osmosis. So that's like our cells. If there's a membrane that's not going to let some things through, then the thing that can move through, water, because there's specific channel proteins for water, then the water moves to make those molecules less concentrated. I'll draw one more picture and then we'll be done. Has everyone gotten this? Yes. Yes? No? Okay. I really wanted that to be even, and it didn't happen. Okay. So if you've got a membrane here that is permeable to water, meaning that it lets water through, so if this membrane is permeable to water, but not permeable to these dots, which direction do you think the water is going to go? The dots can't get through, but the water can. So which side, left or right, is going to be higher or have more water?
Right.
The right side. So the water will move in the direction that will allow an even amount of space between the molecules. So like here, they're all crammed together, and so it's going to move where those molecules can spread out. So oh, sorry. So again, it's going to move in the direction to let these molecules become more diluted. It just wants to add even space between these molecules. Does that make sense? Okay. This is osmosis. We'll talk more about what happens. I feel like I have filled your brain with information. I'm going to let you apply that information that you've learned today before I give you more information. But next time we will talk about what happens to the cells themselves when they're put in different environments. So this is like where we learned about in 10th grade with hypertonic, hypotonic, and isotonic solutions. We'll look into those. The reading on that is still the like 105 through 108, I think it was. So like we didn't get through all of the reading that I had applied or that I had assigned last time. So basically you get a second chance to do that reading.
Maybe I'll read it.
And maybe you'll read it this time. You've got a whole, 4 days to do so. So that way it can be a little more discussion based and a little less lecture based. But we will talk about that next time. What you're going to do with this information. So I have attached on Google Classroom, it's called the membrane transport POGLE. So POGLE is just the company in which that makes these practice problems.